welcome to our exploration of microscopy and cell observation. Over the next 15 minutes, we will delve into the incredible world that microscopes reveal. We'll start with the basics of microscopy, exploring how these powerful tools magnify tiny organisms and structures to make them visible to the human eye. First, we'll discuss the history and evolution of microscopes, from simple lenses developed in the late 16th century to the advanced electron microscopes of today that allow us to see at a nanoscale. We'll highlight essential features such as magnification power, resolution, and the different types of microscopes like optical, electron, and fluorescence microscopes. Next, we'll journey into the world of cells, the building blocks of all living organisms. Using real-time footage, we'll examine various types of cells, including plant cells, animal cells, and bacterial cells. You'll see cell structures like the nucleus, mitochondria, and membrane up close, gaining insights into their functions and importance. We'll also cover the techniques used by scientists to prepare slides and samples for observation, such as staining and fixation, which enhance visibility and detail. These methods have transformed how we understand cell biology, disease mechanisms, and development processes.
By utilizing advanced microscopy techniques.
Have you ever looked at a single drop of water? It seems so simple, so clear. But what if I told you that within that tiny drop, a whole world is teeming with life? A universe of creatures, too small for our eyes to see, is swimming and hunting and living out their entire existence. They are born. They eat. They die in a space smaller than a pinhead. This is not a fantasy. It is a reality that is all around us, in every puddle, on every leaf, even on our own skin. This hidden kingdom contains some of the most bizarre and beautiful life forms on our planet. It is a world of constant activity, full of secrets waiting to be discovered by anyone curious enough to look. We walk through this invisible world every single day. We are surrounded by it, yet we are completely unaware of its presence. Tiny organisms float in the air we breathe. They cling to the food we eat. They build complex communities on almost every surface you can imagine. These microscopic beings are the foundation of much of the life we see around us. They are the architects of our soil. They are the engines of the oceans. Understanding this unseen dimension is not just a scientific curiosity. It is fundamental to understanding our own world, our own health, and our very place within the grand, intricate web of life on Earth. This guide is your invitation to that hidden world. Its purpose is simple. We will learn how to use a microscope, our window to the small. We will explore how to see the invisible life that surrounds us clearly and safely. Think of it not as a complex scientific manual, but as a map for a great adventure. This journey does not require a spaceship or a deep-sea submersible. It only requires a little curiosity and a tool that can bend light. With these, you can become an explorer of a vast and vibrant ecosystem that exists right under your nose. The discoveries you make will be your own witnessed with your own eyes. We will start with the very basics. A microscope might seem like an intimidating instrument full of lenses and knobs, but it is really just a powerful magnifying glass, refined over hundreds of years. We will demystify this tool. I will show you that with a few simple steps, you can learn to control it with confidence. This guide will walk you through preparing your first sample, bringing it into focus, and revealing the details hidden within. We will turn a blurry, confusing image into a sharp, breathtaking view of life at a different scale. Microscopy is, quite simply, the art of seeing the very small. It is the use of an instrument, a microscope, to make tiny objects appear large enough for us to study. This act opens up a universe that is completely inaccessible to our unaided eyes. Without it, the world of cells, bacteria, viruses would remain entirely unknown. Microscopy is our bridge to this invisible realm. It allows us to see the fundamental details of life itself. To understand how a microscope works, We must first understand three important ideas. They are magnification, resolution, contrast. Magnification is perhaps the most familiar concept. It is how much larger the microscope makes an object appear. Resolution is next. Resolution is the ability to show two nearby points as separate. Good resolution does that for tiny structures, turning blur into detail. The third crucial element is contrast. Many cells are almost completely transparent, like ghosts in water. We increase contrast by adjusting light on the microscope or by using special stains. Stains are dyes that color parts of the cell so they stand out. Contrast makes the invisible visible. Not all microscopes are the same. Scientists use different types of microscopes depending on what they want to see. The most common type, and the one you will likely start with, is the light microscope. It uses visible light and a series of lenses to magnify objects. Light microscopes are wonderful for looking at living cells, bacteria, and the tiny creatures in pond water. Another very useful tool is the stereo microscope, which is also known as a dissecting microscope. Unlike a light microscope that shows a flat two-dimensional image, a stereo microscope gives you a three-dimensional view. For seeing the truly, truly small, scientists turn to the electron microscope. They can magnify objects millions of times. With an electron microscope, we can see the exact shape of a virus. Each microscope, therefore, opens a door to a different kind of world. For our journey, we will focus on the light microscope. It provides the ideal balance of power and simplicity to unlock the wonders of the hidden universe all around us. Before we can begin our expedition into the microscopic world, we must gather our tools. First and foremost is the microscope itself, our window to the small. Alongside it, you will need clean glass slides. You will also need cover slips. Next, we need tools for handling our samples. A simple pipette or eyedropper is invaluable. A pair of fine-tipped tweezers will be your best friend. You may also want to have some simple stains. Methylene blue is a classic choice for beginners. It is a safe dye that helps make transparent cells, like those from your own cheek, visible by giving them a rich blue color. First, find a stable, flat surface to work on. Good lighting is also important. Carefully take the microscope out of its box, holding it with two hands. Place it gently on the table with the arm facing you. Always start with the lowest power objective, which is the shortest one, clicked into position directly over the hole in the stage. You will hear a little click as it locks into place. The total magnification is the power of the eyepiece lens multiplied by the power of the objective lens you are using. So, with the 10 times eyepiece and the 4 times objective, you are seeing the object at 40 times its actual size. The larger one is the coarse focus knob, the smaller one is the fine focus knob. Finally, turn on the illuminator. You should see a circle of light appear on the stage. Beneath the condenser, you will find a small lever. This lever controls the diaphragm. Think of the diaphragm like the pupil of your eye. It can open to let in more light, or close to reduce the amount of light. When looking at a very transparent, unstained specimen, closing the diaphragm slightly will increase the contrast. This makes the edges of the cell and its internal structures appear much more defined. For a colorful, stained specimen, you might open the diaphragm to let more light in. That makes the colors appear brighter. Experimentation is key. Before you even place a slide on the stage, take a moment to play with these controls. To begin, take a clean glass slide. Using a pipette or an eyedropper, carefully place a single small drop of your sample liquid in the very center of the slide. To avoid them, hold the cover slip at a 45-degree angle to the slide. Touch one edge of the cover slip to the edge of the water droplet. Then, slowly and gently, lower the cover slip down onto the drop. To clean this up, take a small piece of paper towel or tissue. Gently touch the corner of the paper to the edge of the cover slip. The paper will act like a wick, drawing the excess water away. A single drop of pond water prepared in this way can reveal a bustling community of life. You might see tiny, slipper-shaped paramecia darting about. With your prepared slide on the table, the hunt can begin. Place the slide onto the microscope stage and secure it with the stage clips. Looking from the side of the microscope, turn the coarse focus knob to raise the stage as high as it will go without touching the lens. Now, look through the eyepiece. Slowly, very slowly, turn the coarse focus knob towards you. The moment you see anything at all, stop turning the coarse focus knob. From this point on, only use the fine focus knob to make the image perfectly sharp. When you find something interesting, move the slide so the object is exactly in the center of your field of view. Without touching the focus knobs, carefully rotate the revolving turret to the next objective, the 10x. You might look through the eyepiece and see only a frustrating blur. If focusing does not solve the problem, the next culprit is almost always dirt. To figure out which lens is dirty, try rotating the eyepiece. If the speck of dust spins, then the dirt is on the eyepiece lens. Once you identify the source, you can clean the correct surface using the gentle techniques we discussed earlier. Another common frustration is encountering large dark circles with thick black rings. They are air bubbles trapped under your cover slip. Finally, you might find your image looks washed out. This is an issue of illumination. Slowly close the diaphragm. As you do, you will see the contrast increase dramatically. We must be safe, clean, and respectful in our work. Always wash your hands thoroughly before and after you use your microscope. It is best to have a designated small, sturdy container, like a plastic tub, for used or broken glass. After you are done observing, the most ethical action is to return them to their original environment if possible. This shows respect for the life you have been privileged to witness. Always cover your microscope with its dust cover when it is not in use. Do not be afraid to experiment. Look at everything. Dust from under your bed, mold on a piece of old bread, water from a puddle after a rainstorm. Keep a notebook. Draw what you see. Write down your questions. Never forget the smallest observations can lead to the biggest ideas. Thin slices of cork. Robert Hooke discovered cells. Peering into pond water, Antony van Leeuwenhoek first saw bacteria. Your observations might not change science. They will change you. So go now, look closer. The universe in a droplet is waiting for you.